of protein synthesis. Part 2 is going to focus on mitosis and meiosis. And just so you guys know, this is probably the most biology dense chapter that we'll have this semester, as far as, terms that, may sound a little bit familiar if you've taken high school bio somewhat recently or taken a college bio class somewhat recently. These terms probably sound familiar. But please don't stress if they don't sound familiar, because for this class, really what's important is that we understand the basics. That's why I call this lecture the basics of DNA and cell division, because essentially this is how we inherit genes. This is how we replicate DNA and replicate cells. This is how we code for proteins. So the reason we care about this stuff in a biological anthropology class is because it influences evolution. These processes will help us understand how evolution works, essentially, how the mechanics of evolution works. So that's why we're spending a little bit of time learning about the basics. But please don't stress if this feels a little bit overwhelming. It's okay if it does. But my advice is, if it does feel overwhelming, take a little extra time with it. Watch the recording of the lectures a few times if you need to. Make sure you read the chapter. Make sure you make flashcards for bold terminology throughout the PowerPoints and the reading assignments. Watch the video clips, because it will be to your benefit if you understand the basics of how these processes work. All right, so second half, so part 2 of DNA and the basics of DNA and cell division. We're going to talk about mitosis first. So careful, these two words, mitosis and meiosis, are obviously very similar, but they're very different processes. So mitosis is going to occur in and produce identical diploid somatic cells. So this occurs in our skin cells, our saliva cells, our blood cells, our organ cells, our bone cells. Basically, any somatic cell is produced and continues to go through mitosis throughout your entire life. Now, some cells go through mitosis constantly. Obviously, our skin cells are constantly exposed to environmental stressors, and our saliva cells are constantly, being reproduced. So things like skin cells and saliva cells are constantly going through mitosis. They're going through mitosis as we speak right now. But other cells, like nerve cells and bone cells, don't go through mitosis nearly as frequently. They still do, it's just not as frequent as a skin cell, for example. So that's why if you get nerve damage or if you break a bone, you are capable of recovering, of course. It's just going to take more time. If you get a scrape on your arm, it's going to recover a lot more quickly than a broken bone, for example. All right, so mitosis occurs in and produces identical diploid somatic cells. It involves DNA replication followed by one cell division. It occurs only in somatic cells, only in body cells, and it results in identical copies. So the end result is a parent and a daughter cell, and they are both identical. So the reason it begins with DNA replication is because both the parent cell and the daughter cell will need a complete copy of the genome, essentially, a complete copy of all 46 chromosomes. So the actual division part is broken into what we call prophase, metaphase, anaphase, and telophase. So the interphase portion, which is represented in this picture right here, this circle is interphase. So interphase I like to think of as kind of the prep phase, or the pre-game of mitosis, if you will. So it's basically the cell prepping to divide. So the DNA is replicating and the organelles are replicating, because by the time we get to the end, both cells, both parent and daughter cells, will need their own organelles and also their own DNA, their own chromosomes. During prophase, that's the first phase of actual division. During prophase, the chromosomes are forming. So DNA is coiling around these proteins that we call histones. It's coiling around these histones to form visible chromosomes. And also the spindle apparatus is forming. So that's prophase. So the way I remember it, pro means first. So prophase is the first phase of actual cell division. The middle phase is what we call metaphase. So the way I remember it is middle meta. And also, conveniently enough, during metaphase, all of the sister chromatids, the paired up chromosomes, are going to be aligned along the equator or the middle of the cell. So you're seeing all these little X's and they're lined up along the equator of the cell. And the spindle apparatus is what's pushing them into that positioning. So the spindle apparatus is pushing the sister chromatids into the equator or middle of the cell. The fourth phase, or fourth step, I should say, the fourth step in mitosis is called anaphase. And the big event during anaphase is something called disjunction. So during disjunction, the spindle apparatus is actually ripping apart the sister chromatids at the centromere and pulling them to opposite poles of the cell. Okay, so if my fingers are chromosomes, the end result of anaphase will look like this. Okay, we've got all the chromosomes are now in single form, and they're pulled to opposite poles of the cell. And then the very last phase, the fifth step, is called telophase, or also known as cytokinesis. So during telophase or cytokinesis, the cleavage furrow pinches in and forms two identical diploid cells. So our end result of mitosis, we've got 46 chromosomes in the nucleus. I mean, I know there's only 4 in this picture, but so envision that there's 46 chromosomes located in the nucleus of each the parent and the daughter cell. And other cool thing, once we finish telophase, it goes right back into interphase. Okay, so our cells are constantly going through mitosis. It's a constant, continual process our cells go through.
Excuse me.
So,
I'm sorry.
yes, Loretta.
I missed the third phase. What was the third phase again, please?
The third phase is what we call metaphase. So the way I remember it, I think middle is the way I remember it. And I also remember that during the middle phase, during metaphase, the sister chromatids, so the chromosomes that are all paired up like this, the sister chromatids are going to be aligned along the middle or the equator of the cell, essentially along this middle part.
Okay, I think I missed the first phase then. I had that one.
Okay, the first phase is what we call, well, the first phase, the prep phase is called interphase.
Okay, I got it.
So
I got everything now. Thank you so much. I just needed that.
Perfect.
First.
Of course, you're very, very welcome. Good question, Laredo.
Okay.
Any other questions?
Can you quickly just say all the phases again, please?
Sure. Yeah. So interphase is the pregame. It's the pregame of mitosis. So all the DNA is replicating and the organelles are replicating. That's called interphase. Then we go into prophase. So that's the first phase of actual division. So during prophase, the DNA is coiling around those proteins we call histones to form visible chromosomes. And then after that, we've got metaphase. So during metaphase, all of the sister chromatids are aligned along the equator of the cell. So think meta, middle. And then the fourth step is what we call anaphase. So during anaphase, that's when the disjunction happens. That's when the spindle apparatus is ripping apart the sister chromatids and pulling them to opposite poles of the cell. We call that anaphase. And then the very last step, the fifth step, is called telophase, also known as cytokinesis. So that's essentially the conclusion of cell division. The nuclear membrane is pinching in to form two identical cells. So you've got the original parent cell that still has its 46 chromosomes and all of its organelles. And then you've got an identical daughter cell that also has its own 46 chromosomes and own individual organelles. So the parent cell and daughter cell are going to be identical.
Okay, thank you so much.
You're very welcome. Excellent questions. Okay, so just some quick points about mitosis, essentially why we care. So mitosis is critical for growth and tissue repair. Without mitosis, we'd be in big trouble because obviously we get scrapes on our arms and break our bones and need to replace our blood cells and saliva cells all the time. So without mitosis, we as organisms would not be able to do that. So mitosis begins immediately at zygote formation, essentially. So the second the sperm fertilizes the egg, mitosis will begin. Cell division continues and cells will become specialized to perform special functions, specific functions. So some cells become tissues, some become organs, some become bone cells, blood cells. You guys get the picture. So mitosis occurs in your cells throughout your life. Although remember, some cells go through mitosis more frequently. Your skin cells, for example, are constantly replicating. But nerve cells are ones that don't go through mitosis very frequently. That's why it can be so catastrophic, so devastating when you get nerve damage, because even though it is possible, you can certainly recover from nerve damage. It takes time and it takes a lot of sometimes outside medical intervention to recover from nerve damage. All right. Okay, so many cells, such as skin cells, are constantly replaced. Other cells, such as nerve cells, are rarely replaced. The entire process, if you're looking at interphase, prophase, metaphase, anaphase, telophase, all 5 steps take place in about 18 to 24 hours. But interestingly enough, the vast majority of the time, About 18 to 23 hours, your cells are in that interphase, the pregame. There's a lot of pregaming going on in mitosis. So they're doing lots of DNA replication. They're replicating the DNA and they're replicating the organelles. The actual division process, the prophase, metaphase, anaphase, telophase, that process only takes about an hour. So if we had to guess, if we were, in Vegas and we wanted to guess how many of our cells are in each phase, it would be the most likely we should put our money on interphase, because the majority of our cells, like right now, are probably in interphase. All right, how are we feeling about mitosis before we watch a short clip on it? Do we have questions about mitosis? Okay, so I'm going to show you guys a little clip on mitosis. Now these are produced by something called Amoeba Sisters. And please know I'm not, these may seem a little bit childish. Please know I'm not trying to insult your intelligence. I've just found that for processes like these, it actually is really helpful to just break it down and make it really easy. So please know I'm not trying to insult your intelligence. I just have found that these Amoeba Sister videos are actually really helpful, especially if, because we're not in a bio class or an anthropology class. I don't want to make it too overly complicated for you.
Oh,
All
no,
right, so let's go ahead and watch it. I'm sorry. Yes, go ahead, Lorena.
No, I said, don't be worried. I'm excited to see it.
Perfect. Perfect. All right, so let's go ahead and we're going to watch the video clip. Obviously, if you just sit back and relax and enjoy it, but if you think of a pressing question, feel free to write it down and we'll talk about it after.
Have you ever looked down at your leg or your arm to find some cut and you have no idea where it came from or how you got it? So you put a bandage on it and a few days later it's gone. It's all healed and you don't even think anything of it, of this amazing process that causes this to happen. Or let's say you're looking at your nails and you notice they're a lot longer since the last time you cut them. Or let's say you're looking in the mirror and you notice that you are a lot bigger than you were when you were 5 years old. What do all of these things have in common? Well, one major thing they have in common is mitosis. Mitosis is a type of cell division done by most of your body cells, and it's really important for cells to divide. If they didn't divide, you wouldn't grow. I mean, how do you grow if you can't make more cells, right? So one reason why you're bigger than you were when you were 5 is mitosis. Mitosis also is great for repair of damage. If you have some kind of accident, like when we were talking about that cut on your arm or leg, well, you want to make sure it gets repaired well. So you have to make more cells to do that. Mitosis is great for that. Now, it's really important to understand what it is not. Mitosis is not a process that makes sperm or egg cells, because that's something different called meiosis, which sounds like mitosis, unfortunately, but it's a different process. Mitosis is done to produce body cells. Mitosis makes identical cells. That is the goal, identical cells. So if you're trying to make more skin cells to replace worn out or damaged skin cells, you don't want to start suddenly making stomach cells there. That would be ridiculous. You want to make sure you have identical cells replacing what was lost. So mitosis makes identical cells. It's a really important thing. Now, it's also important to understand that your cells are not dividing all the time. If all they did was divide, it would just be rapid, crazy growth. In fact, this is kind of what cancer is. Cancer is uncontrolled cell growth. We have a clip on the cell cycle and what the cell is usually doing most of its daily life, which is actually a phase called interphase, where it's growing and replicating its DNA and carrying out its daily cell functions. That's where cells spend most of their time in respect to the whole cell cycle. Mitosis is a very short amount of time in respect to the whole cell cycle. But mitosis is a critical process because this is where it is going to divide and make more cells. Before we get into the steps of division, it's really important to understand that your cells have something inside them, an organelle called the nucleus. And the nucleus holds your DNA. DNA is really important because it's your genetic information. And if you're going to make more cells, you need to have the same DNA in those new cells as you did in your original cells. You want it to be identical, no mistakes. Very important. The problem is you've got a lot of DNA, And we've got to get that DNA into the new cells using mitosis. So there has to be a better way to organize that DNA. Well, what actually happens is that DNA can be organized into these condensed units called chromosomes. Chromosomes are made of DNA and protein. You've probably heard before that humans have 46 chromosomes. That means 46 chromosomes are found in most human body cell nuclei. What are nuclei? Well, it's the plural of nucleus. You don't say nucleuses, you say nuclei. Well, in the nuclei, there are 46 chromosomes. Organizing DNA into condensed chromosomes makes it a lot easier to move over when you're making new cells. So if you have 46 chromosomes in a human body cell, you have to duplicate those chromosomes in interphase before mitosis starts. That basically means you're duplicating your DNA, since chromosomes are made of DNA and protein. You have to do this before mitosis starts because if you're going to make an identical cell that has 46 chromosomes, just like the original, well, it makes sense you have to duplicate the genetic material before splitting. So if you look at our cell cycle video clip, we talk about interphase. That's a stage where most of the time cells are spending their time. They're actually duplicating their DNA during that time. So ready for the tricky part? Well, because we tend to count chromosomes by the number of centromeres present, when the 46 chromosomes duplicate, we still say there are 46 chromosomes, as the sister chromatids are still attached, and we're counting by centromeres. So 46 chromosomes here, they replicate in interphase, and you still have 46 chromosomes in this picture, but you went from 46 to 92 chromatids. We have a video explaining that in more depth and how that factors in for mitosis. Okay, so now we can get right into mitosis. I like to tell students to remember PMAT. It's a little acronym that helps you remember. The P is for prophase, the M is for metaphase, the A is for anaphase, and the T is for telophase. So remember PMAT, the stages in order. The very first step is prophase. Prophase because it's the beginning step, the nucleus is still there, and it's going to go away later on, but this is a stage where it's actually still there. The chromosomes are visible. In fact, we say they're condensing, which means they are thickening and visible. The next stage is metaphase. M for metaphase, but I also like to remember M for middle because in this stage, the chromosomes line up in the middle of the cell. The nucleus has been disassembled, it's no longer there, so we've got the chromosomes in the middle waiting there. Next, the A is for anaphase. In anaphase, I like to think as the A for away. The chromosomes move away. They are moving to opposite sides of the cell, so they are moving towards the poles of the cell. Now, one thing to point out, these chromosomes, they're not moving by themselves. They actually have something called spindles. These spindles are fibers that help move the chromosomes to the ends. Kind of helps them move along. The last stage of mitosis, think T for telophase. In telophase, the chromosomes are actually at the complete opposite ends, and new nuclei are forming on each side to make these two new cells. The nuclei are starting to surround the chromosomes on both sides. I like to think of the T for 2 because you can really see in this step that the end goal is going to be 2 cells, and in the human body, they're each going to have 46 chromosomes. And again, remember, they are identical. Now, cytokinesis is responsible for the final separation into 2 cells by splitting the cytoplasm, which completes after the PMAT mitosis stages. So why did all this matter? Without understanding cell division, we wouldn't understand how growth and repair happens, because they both require more cells to be made. Understanding mitosis is also very important for cancer research. Cancer itself is uncontrolled cell growth. So, in other words, uncontrolled mitosis. Well, that's it for the Amoeba Sisters, and we remind you to stay curious.
All right, so that was our nice Amoeba Sisters summary of what we just talked about with mitosis. So remember, mitosis is always going to be the production of identical somatic cells that are diploid. Okay, so if they're diploid, that means in the chromosomes, or excuse me, within the nucleus, how many chromosomes would we find in a diploid cell?
46.
46. Excellent. So every single cell that's produced by mitosis is going to be identical to the original cell, and it will have its own set of 46 chromosomes. So the production of identical diploid daughter cells. Okay, so let's shift gears and let's talk about meiosis. So again, like Amita Sisters pointed out as well, meiosis and mitosis are very similar words. So just be careful when you're reading exam questions or learning about the topic in general. Just make sure that you fully look at the word carefully and understand whether it's talking about mitosis or meiosis. So meiosis will be the production of unique gametes that are haploid. So it actually begins, and we'll talk about this here in a moment, but it's going to begin with a specialized somatic cell that will go through 2 cell divisions. So it's 1 DNA replication followed by 2 cell divisions. So there's just one interphase, but then there's a prophase. There's a prophase 1, metaphase 1, anaphase 1, telophase 2. And then there's a whole nother process. So it doesn't go through interphase again, but it will go through prophase 2, metaphase 2, anaphase 2, and telophase 2. Okay, now I'm not going to require that you know the specifics of each of those phases, but I do want you to know that it is 1 DNA replication followed by 2 cell divisions. And I do want you to know that it produces gametes, so it produces unique sperm and egg cells. Ova is another word for egg. So your end result is 4 haploid daughter cells that have 23 chromosomes each. And I do want you to know that it produces unique gametes. So essentially every single human, every female, every male has a capacity to produce over 1 million different unique gametes. So that's why even if you have the same biological mother and father as your siblings, you don't look identical. You may look similar to your siblings. But for example, I look nothing like mine. I look nothing like mine at all, but we have the same biological mother and father. So that is certainly possible because every single human has the capacity to produce over 1 million different unique gametes. So it's important to remember that mitosis, that we talked about before, mitosis is the production of identical diploid cells, but meiosis is the production of unique haploid gametes, okay? So let's talk about how it differs in males versus females. So for males, it's going to occur in the gonads, which is the testes, and it begins with a specialized somatic cell called a primary spermatocyte. So the primary spermatocyte is a specialized somatic cell that's found in the testes of males, and it will divide to produce genetically unique sperm cells. So we'll say that again. The primary spermatocyte is a specialized somatic cell that's located in the testes of males, and it will divide to produce genetically unique sperm cells, okay? In males, the production of sperm cells will begin at puberty and it will end at death, okay? Now, of course, males go through a similar process to menopause, so it's similar to what females go through. So when they hit a certain age, the production of sperm cells certainly slows down due to changes in testosterone. The sperm count will decline, but essentially in males, the process begins at puberty and then will end at death. The sperm cells are produced and stored in the epididymis for 10 to 20 days, where they're either then used or reabsorbed by the body. Okay, so that's meiosis in males. So it's a little bit different for females. So for females, similar to the primary spermatocyte, we also have a specialized somatic cell that's found in our gonads, found in the ovaries. It's called the primary oocyte. So the primary oocyte is a specialized somatic cell found in the ovaries of females that will divide to produce genetically unique egg cells or ovas. So for females, so if you, and next time you see your grandmother, if your grandmother's still alive, you should tell her this neat little bit of information. So you were actually carried not only by your mother, but also your grandmother. Because when your mother was still in your grandmother's womb, she was already producing her egg cells about 12 weeks after fertilization. So when your mother was still in your grandmother's womb, she was already producing her egg cells. So every female is born with all of the egg cells they will ever have. So all of us were carried not just by our mother, but also our grandmother. We were just little ovas, little egg cells when we were in our grandmother's womb, but we were there. So I'm sure if anybody's grandmothers are still alive, I'm sure they would love to hear this story. So in females, the production of egg cells begins before birth, beginning about 12 weeks after fertilization. But those egg cells obviously they're not active yet. Those egg cells, those ovas, are going to be held in what we call suspended animation until that female hits puberty. So the onset of menstruation is called menarche. Menarche is when menstruation begins, when that 28-day cycle begins for females. So during that process, it's a 28-day cycle. And during that process, the eggs are, if they're not fertilized, then they are released through menstruation. So generally the middle of the cycle, generally days 12 through 16, are when that egg could potentially be fertilized by a sperm cell. Okay. But essentially most women go through this 28-day cycle. It may vary by a day or two, or even a few days or two, but in general, this 28-day cycle begins at menarche, the onset of menstruation, and will continue all the way until that female hits menopause, or the cessation of reproduction, essentially. So for females, there's a little bit more of a marked beginning and end to reproduction. Okay, it begins with menarche, and then it ends at menopause. Okay, is this making sense? Yes, a little bit of sex ed for you guys today. All right, do we have questions about meiosis?
No, I just think it's, well, yes, but no. I think it's really interesting that you made the comment about how the grandmother passes down that egg process to the mom, because I look a lot like my grandmother. So it's just interesting to discover that. Thank you for enlightening me.
Isn't that cool? Yeah,
Absolutely.
Isn't that, when I first learned this, I told my grandmother and she thought it was the coolest thing ever. So I wanted to share that little bit of information because I know that mine really loved it.
I will share with my mom. I wish my grandmother was here to share with her, but my mom, I'll definitely let her know.
Yeah, my mom loved it too. It's a cool bit of information that, even though some of this stuff may seem a little bit, learning the details, but there's some cool little bits of information that, once we understand biological processes, these cool little bits start to come out. So I'm glad you guys are enjoying it.
Okay.
All right, other questions about meiosis before we watch a little video clip on it? Okay, so we got Amoeba Sisters again to kind of give us a summary. So my advice with these, just kind of sit back and enjoy. Obviously, if a pressing question comes up, feel free to write it down just so you don't forget to ask it. But my advice is just kind of sit back and enjoy and take it all in. Oops, that's not what I meant to do. I meant to hit play.
Have you ever wondered how two siblings can have the same mom and dad and still look so different? Well, today we're going to talk about a process that makes that possible, a process called meiosis, not to be confused with mitosis, which sounds unfortunately similar. Mitosis makes identical body cells, like your skin cells and stomach cells. Recall from our mitosis clip that since it makes identical body cells, mitosis is important for growth and for repair of damage, or to replace worn-out cells. But not meiosis. Meiosis is a process that contributes to genetic variety. Meiosis also doesn't make body cells. It makes sperm and egg cells, otherwise known as gametes, the fancier word. You might recall that humans have 46 chromosomes. That's how many chromosomes most body cells in your body have. But there are some human cells that don't have 46 chromosomes. Human sperm cells and egg cells have 23 chromosomes. Why the number difference? Well, if a sperm cell has 23 chromosomes and an egg cell has 23 chromosomes, when they come together, that makes 46 chromosomes. That will allow a newly formed fertilized egg to develop into a human. Meiosis is what we call a reduction division because you have a starting cell that has 46 chromosomes, and your ending cell, the sperm and egg cells, have only 23 chromosomes. Before we can start getting into the stages of meiosis to make gametes, we have to remember what happens before meiosis can even start. Actually, this also happens before mitosis. It's the stage known as interphase. And if you remember interphase, it's when the cell is growing, it's replicating its DNA, it's carrying out cell processes, and just like mitosis, interphase happens before meiosis is even going to start. So the starting cell has 46 chromosomes. And you have to duplicate those chromosomes in interphase before meiosis starts. That basically means you are duplicating your DNA, since chromosomes are made of DNA and protein. Ready for the tricky part? Well, because we tend to count chromosomes by the number of centromeres present, when the 46 chromosomes duplicate, we still say there are 46 chromosomes, because the sister chromatids are still attached, and we are actually counting by centromeres. So 46 chromosomes here, they replicate in interphase, and you still have 46 chromosomes in this picture. But you went from 46 to 92 chromatids. Little tricky there. We have a detailed video that explains these chromosome numbers before and after replicating in interphase that can be useful for meiosis. Okay, so interphase checklist done. Now we can move into meiosis. You might remember the mitosis stages PMAT. The P was for prophase, the M was for metaphase, the A for anaphase, and the T for telophase. And the good news is that in meiosis, you still use those terms. But because meiosis is actually a reduction division, you're going from 46 chromosomes to 23, which means you actually divide twice. So instead of mitosis, where you divide one time and do PMAT one time, in meiosis, you're going to divide twice and therefore do PMAT twice. And because of this, in meiosis, you put numbers after the phases to indicate whether you're in the first division or the second division. So let's dive right in. So let's start with the very first step, prophase 1. One thing I like to remember about prophase is pro, this actually means before, and it kind of helps you remember that it comes before all the other stages start. This is where the chromosomes are going to condense and thicken. They are actually going to line up with their homologous pairs. The word homologous means that the chromosomes are approximately the same size and they contain the same types of genes in the same locations. They're going to match up. It is during this prophase 1 that this amazing process occurs called crossing over. Now, I know crossing over probably sounds like something very different, but this is actually a really awesome process because this is when the chromosomes, they're lined up in homologous pairs, and they have this way that they can transfer their genetic information and exchange it between each other. It's kind of like these chromosomes flop over each other, and they do a little genetic information exchange here. It makes for what we call recombinant chromosomes, which can eventually contribute to the variety that we were mentioning that siblings can have even when they have the same parents. More about that later. Now we move into metaphase 1. In metaphase 1, think of the M as standing for middle. The chromosomes are now going to be in the middle of the cell. It's a little bit different though from mitosis because these chromosomes are going to be in pairs in the middle of the cell. So it's not a single file line, they're in pairs in the middle. Now during anaphase 1, think of A for away because the chromosomes are going to be pulled away by the spindle fibers. Then we end with telophase 1, where you have two newly formed nuclei, and it becomes obvious that you're going to end meiosis 1 with two new cells. Cytokinesis follows with splitting the cytoplasm. But we're not done yet. On to meiosis 2. The very first step in meiosis 2 is prophase 2. It's not going to be nearly as eventful as it was in prophase 1, though, because they're not going to have homologous pairs of chromosomes. They also are not going to have this amazing process called crossing over. That doesn't happen again in prophase 2. You have your chromosomes, and the spindles are starting to form like they did in prophase 1, but prophase 2 is just not as eventful of having that process of crossing over. In metaphase 2, remember, think M for middle, the chromosomes are going to line up in the middle. This time, though, they are in a single file line. They are not in pairs like they were in metaphase 1. Anaphase 2, remember A for away, but this time it's the chromatids that are going to be pulled away by the spindle fibers. In telophase 2, you can see the nuclei reforming, and you can also see that the two cells have divided. There's going to be 4 cells forming. Cytokinesis will follow to completely split the cytoplasm. Now, keep in mind that meiosis in males produces sperm cells, and in females it produces egg cells. Because of independent assortment and also crossing over, you're going to have variety. For example, in a male, the 4 sperm cells that are produced each time, they are all different from each other. They're also different from the starting cell because the starting cell had 46 chromosomes, and the ending cells only had 23. So they are not identical to the original, and they are not identical to each other. This is going to lead to variety, a reason why 2 siblings with the same parents can look different from each other. They still developed from a unique egg and a unique sperm cell that came together. One last thing to think about. Scientists are often looking into the process of meiosis because sometimes the chromosomes don't separate correctly. It's called non-disjunction when a cell can receive too many or too few chromosomes in the separation. This contributes to some genetic disorders, which is something scientists continue to study. Well, that's it for the Amoeba Sisters, and we remind you to stay curious.
All right, that was our little summary from the Amoeba Sisters on meiosis. So let's look at these two processes right next to each other, just to make sure that we understand the differences. So this is mitosis right next to meiosis. So thinking back to the beginning of this lecture, mitosis is the process that produces identical somatic cells. So this occurs in and produces identical somatic cells. So our skin cells, our organ cells, our blood cells, our tissue cells. The function, essentially why we care, why it's important to us as anthropologists. We care because without it, we wouldn't have growth and tissue repair. The end result is two identical diploid cells. And this process occurs throughout our body, beginning at fertilization or zygote formation, and then ending at death. But of course, some cells go through mitosis more frequently than others. Skin cells, for example, go through mitosis constantly. Nerve cells hardly ever go through mitosis. The second process that we talked about, the second type of cell division, is called meiosis. So meiosis is going to begin with those specialized somatic cells. So either the primary spermatocyte or the primary oocyte. Primary spermatocyte for males, primary oocyte for females. And it will go through meiosis 1 and meiosis 2, and crossing over an independent assortment will create those genetically unique gametes, genetically unique sperm and egg cells. So the function, essentially why we care as anthropologists, we care because without it, we wouldn't have reproduction, obviously, and we wouldn't have the genetic variation and variability necessary for evolution to occur. So our end result is 4 non-identical, or essentially 4 unique haploid cells. And it only occurs in the gonads of males and females. So for females, it occurs entirely in the ovaries. For males, it occurs entirely in the testes. All right, questions about mitosis versus meiosis? Anything still fuzzy? All right, feeling good. All right, let's move on. So we've already talked about chromosomes. So chromosomes were introduced in part 1 of this lecture. But in humans, obviously, we have 46 chromosomes, or 23 pairs in the typical condition. Pair number 1 through 22, we call those the autosomes. The 23rd pair will determine our biological sex. If we have 2 X chromosomes, we are biologically female. If we have an X and a Y chromosome, we are biologically male. Now, there are some aberrations and abnormalities upon that, though. So even biologically speaking, technically, there are more than 2 sexes. And I'll tell you why here in a moment. So we know that mistakes can occur during meiosis that will result in genetic abnormalities or aberrations, we like to call them. So a chromosomal mutation is a mistake during meiosis that results in an extra or missing piece of a chromosome, an entire extra or missing chromosome, or even an entire extra set or missing set of chromosomes. So a chromosomal anomaly would be having too many or too few chromosomes. So a well-known example would be what we call trisomy 21. So when you break that word up, tri means 3, somy means chromosome. So what happened in a trisomy, that means there was a non-disjunction, there was a failure of the chromosomes to separate at the 21st location. So what that resulted in is either an egg cell or a sperm cell that had an extra 21st chromosome. So then once it combines with the other gamete, so let's say the egg cell had the extra 21st chromosome, once it combined with the sperm cell, we ended up with 23 of the 21st position, you ended up with 3 of the 21st chromosome. So that's also known as Down syndrome. So chromosomal aberrations, they're similar, but they're so severe that the body essentially recognizes that fetus as unable to survive, and it usually results in early miscarriage. Okay, so non-disjunctions will occur when the chromosomes fail to separate properly during cell division. So they'll be the most detrimental if they occur during meiosis, because that means that there's egg cells and sperm cells out there with the wrong number of chromosomes. with either 24 or 22 when we need 23 in the typical gamete. So if a non-disjunction occurs during meiosis, then the resulting gametes will have the improper number of chromosomes. So instead of the 23, they would have either 24 if they have an extra, or they would have 22 if they have one missing. So the standard condition, so essentially what's the typical, what most humans have, is called disomy. So break up that word. Di means two, somy means chromosomes. So that's the typical condition. We have two chromosomes. We have pairs of chromosomes at each location. We have 22 pairs of autosomes and 1 pair of sex chromosomes. The next two are going to be the aberrations or the abnormalities. A monosomy, break up that word. Mono means one, somy means chromosome. So a monosomy occurs when one pair is missing. So this example here is a monosomy that's occurring on the 23rd pair on the sex chromosomes. So this individual has XO syndrome. So they have one X chromosome, but they are missing the second X chromosome. So this individual, generally, those that have Turner syndrome, they have mainly female sexual characteristics. They sometimes are sterile. They sometimes have heart malformations. They have some other aspects to their disorder as well. But monosomies are survivable. It is possible to live a completely healthy life with a monosomy. Trisomy is essentially an extra one. So break up that word. Tri means three, somy means chromosomes. So trisomies are chromosomal aberrations or abnormalities that occur when there's an extra chromosome present. So those are the cases where there was an extra, so you end up with a sperm or an egg cell that has 24 chromosomes instead of 23. So once you combine, you end up with either a trisomy 21, trisomy 11, or trisomy 18, or three of the most common ones. Now thinking back to what we learned about karyotyping. So this is in the part 1 lecture. So karyotyping, that's when we're looking at a picture of all of the chromosomes lined up in order. Let me find that picture for you guys so I can show you what I mean. Let me see here. Let's look at this picture up here. Okay, there it is. Okay, so this is our karyotype. So it's a picture or a representation of all 46 chromosomes, all 23 pairs. So this is a picture of a typical female. So we've got 1 through 22, those are her autosomes, and then she's got 2 X chromosomes. Now, if you guys are noticing, the homologous chromosomes are identical. They've got the same banding pattern, they've got the same centromere location, they're the same length. So that's why they're homologous, because at each location, they're coding for, those are the alleles for a similar characteristic. Now, the larger the chromosome, the more genes. So essentially, chromosomal aberrations that occur on chromosomes 1 through 10 are simply too severe for survival, because the larger the chromosome, the more genes. So typically, it's extremely rare for an organism or human to survive if there's an aberration on chromosome 1 through 10. Now, the smaller the chromosome, the fewer the genes are located on that chromosome. So that's why trisomy 21 or Down syndrome, even though it has severe implications for that human, it is one of the most survivable ones as far as if that organism can survive and have a productive life. Okay, so trisomy 21, even though it has some implications, that's one of the most survivable ones. Okay, there's also trisomy 11, and I always mix these two up. One of them is Edwards syndrome and one of them is Patau, but trisomy 11, I believe, is Edwards, and then trisomy 18 is Patau syndrome. So those are 3 common chromosomal abnormalities that can occur in humans. But some of those chromosomal aberrations are simply too severe for survival. So chromosomal aberration will oftentimes result in an early miscarriage. Okay. All right, how are we feeling about chromosomal aberrations? Any questions about that? We're feeling good? Okay. All right, let's talk genes. So just like there's types of cell division, just like there's classes of cells, there's also classes of genes. So some genes are structural and some are regulatory. So the structural genes, just like they sound, are responsible for bodily structures. So they code for things like whether or not we have attached or free hanging earlobes, whether or not we have a straight hairline or a widow's peak, whether or not we have type A blood, type B blood, or type O blood. Regulatory genes will turn other genes on and off. So you may also hear them called Hox genes or master genes. So these genes influence the way that our structural genes are expressed, essentially. All right, so this is a little bit of a review because we already did Punnett squares, but just to review, each gene will have a specific location on the chromosome, and we call that a locus. So that term locus just means the location of the gene on the chromosome. So loci are valuable in understanding genetic variation, and the alleles on the various loci may be chemically identical. If they're chemically identical, we call that homozygous. Chemically identical is homozygous, and then chemically alternative versions of the same gene are always called heterozygous. So in this example here, we're looking at plant color. So we've got the homologous chromosomes right here. So at this locus, so this is the locus that's coding for the color of the plant. So from one parent, this plant inherited the allele for purple flowers, and from the other parent, this plant inherited an allele for white flowers. Now, if it's simple monohybrid dominant recessiveness, whichever allele is dominant will end up being the color of the plant. So if purple is the dominant allele, then those flowers will be purple. And if white is the dominant allele, then those flowers will be white. Okay, but some genes are more complicated than that. Some genes are what we call polymorphic and also pleiotropic. We'll talk about pleiotropic in a minute, but just to avoid confusion, poly, so break up that word poly means many. So polymorphic means a characteristic that's influenced by multiple genes at multiple different loci. So skin color, eye color, hair color, those are all polymorphic traits. So they're determined by genes at multiple locations on chromosomes, and they're also influenced by environmental circumstances. And we'll get into that in more detail in chapter 5, actually. This slide here is actually completely review. You've already written this information down, so I'm not going to spend much time on it, but just to review, a genotype is the genetic makeup of an organism. The phenotype is the physical expression of the genotype, and it may be influenced by the environment. The different types of genotypes, so chemically identical alleles are called homozygous, and there's 2 types of homozygous. Homozygous dominant would be 2 copies of the dominant allele. So we always represent homozygous with 2 uppercase letters, or the only other possibility for homozygous would be homozygous recessive. So homozygous recessive is always represented by 2 lowercase letters. Your third possibility for genotype would be chemically alternative or different alleles. So if you inherit one dominant allele from dad, for example, and a recessive allele from mom, so one dominant big A, one recessive little a, the combination of that is called heterozygous. All right, so ABO blood typing. So we've done some Punnett squares with this, and now we're going to talk a little bit more about how it works as far as who can donate to whom. So human blood type is a genetic trait with different alleles. It's an example of a polymorphism, and it's also an example of codominance. So each person will have one of 4 blood types. So these are the phenotypes. So you could either be blood type A, you could be blood type B, you could be blood type AB, or you could be blood type O. So the combination of those alleles that you inherit from your parents will determine your overall genotype and phenotype. So each person has either an A, B, or an O allele on one chromosome inherited from one parent. So let's say the first one is inherited from mom, for example, and then each person will have either an A, B, or an O allele that's inherited from dad. And the combination of what you inherit from mom and dad will determine the person's blood type. So if you're blood type A, you actually have 2 possible genotypes. You could have inherited an A allele from both mom and dad, or you could have inherited an A from mom and a recessive O from dad, for example. But either way, as long as you have at least one big A, because that's the dominant allele, as long as you have at least one big A, your overall blood type is type A blood. Same deal for type B. So 2 possibilities for your genotype. You could inherit a type B or a B allele from both mom and dad. So BB also results in type B blood, or your second possibility, you can inherit a B allele from dad, for example, and an O allele from mom or vice versa. But as long as you have at least one dominant B, your overall blood type is still type B. The codominant condition, the type AB blood, because both A and B are dominant, if you inherit an A allele from mom and a B allele from dad, or vice versa, that combination will make your overall blood type type AB. Okay, that's the codominant condition. Now, the only way you end up with type O blood, because O's are recessive, the O allele is recessive, you must inherit an O allele from both mom and dad. So if you get an O from mom and an O from dad, that combination, OO blood, overall phenotype is type O. Okay, so let's talk about who can donate to whom and who can receive from whom. So for example, if you're going to receive a blood transfusion, you have to make sure, or the doctor has to make sure, that your blood type is compatible with the transfused blood. So for example, if you have type A blood, that means on the surface of your red blood cells, you have something called antigens that identify your red blood cells as type A. Okay, so if you have type A blood, you can get type specific. You can obviously get a donation from another human that has type A blood, or you could receive a transfusion from the universal donor, which is type O. Type O blood, but does not have anti-A or anti-B, excuse me. It does not have anti-A or anti-B on the surface of its red blood cells. Okay, now let's talk about what happens if you get a donation from somebody that does have an opposing antigen. So let's say, again, let's pretend I have type A blood, and let's say that my doctor doesn't check and he tries to give me a donation from somebody with type B. So on my red blood cells, I have the A antigens. On the donor's red blood cells, he or she has the B antigens. So what's going to happen as soon as I get that blood transfusion, my body is going to recognize that blood as foreign, and it's going to start to fight it off. There's going to be what we call an antibody antigen reaction that we call agglutination. I always have a hard time with that word. It's called agglutination here on this slide. So that agglutination is essentially the antibody antigen reaction of my red blood cells, my A antigens, recognizing the B antigens as foreign and there's an autoimmune response, and it's trying to fight them off. Okay, so essentially what happens, the antibodies will attach themselves to the foreign antigens, causing clumping of the red blood cells, and the clumped red blood cells will be unable to pass through the blood vessels. So that clumping and clotting will prevent red blood cells from reaching your vital organs. So our red blood cells carry oxygen. Our organs need oxygen to function and survive. So essentially, if you get a donation from somebody with a non-compatible blood type, unless your doctor takes quick action, this is something that could be deadly. It could result in organ failure and possibly even death without quick medical intervention. Okay, so if you have blood type A, you can receive a transfusion from somebody else that has type A or from type O. If you have blood type B, you can receive a transfusion from somebody with type B blood or somebody with type O. If you have type AB, you're the universal recipient. So you can get obviously somebody else's blood that also has AB, or you can get a donation from type A because it will recognize the A antigens. You could get a donation from somebody with type B because you will recognize the B antigens, or of course you can get O because there are no antigens. Now, if you have blood type O, you can only receive from other people that have type O. So even though you can donate to anyone, you can only receive from others that have type O. All right, how are we feeling about that? You feeling good? Okay. So last slide. So I mentioned that many genes are far more complicated than that Mendelian one gene, one protein model. We did Punnett squares, and we talked about several things. We talked about earlobe form, we talked about tongue rolling, we talked about widow's peaks, we talked about hitchhiker's thumbs, we talked about PTC tasting. So there's, about 10 traits that are monohybrid. But most human characteristics are far more complicated than that. Many traits are polygenic, and they are determined by genes at more than one locus. So remember that term locus, that vocab word locus means the location of the gene on the chromosome. So most of our characteristics are going to be polygenic, meaning that they are determined by genes at 2 or more loci, or 2 or more locations on chromosomes. So in addition to being polygenic or influenced by multiple genes, most polygenic traits are also influenced by the environment. So a great example, the pigmentation in our skin, whether or not we have eumelanin or pheomelanin, is influenced by multiple genes at multiple locations on chromosomes. And it's also influenced by our UV exposure, okay? So I don't want to give too much away on that because chapter 5 goes into this in detail. In topic 5, we'll go into this in detail. But just for now, know that many of our genes, or many of our characteristics, I should say, many of our traits, many of the things that we can see are actually influenced by more than one gene, more than one protein. Okay, so that's why sometimes it can be very complicated to understand human genetics. All right. How are we feeling? Questions? I'm going to stop the recording.